Welcome to Biology 1408, Chapter 3, Cell Structure and Function from the Concepts and Connections textbook by OpenStax. This chapter will cover topics about various types of cells and the functions of components within the cells. By the end of this chapter, you will be able to describe the differences between various types of cells, functions of components within these cells, and how substances move in and out of the cell. In order to see a cell, you must use an instrument, which magnifies the cell to be visible by the eye. The first microscope was invented in 1590, but it wasn't until the 17th century that the scientist Robert Hooke modified the microscope and identified the first cell. Today, there are many different types of microscopes, all of which are used for different purposes. The light microscope on the left is used to magnify small specimens and can be used to visualize cross-sections of samples or even living organisms, although they must be small enough to fit on a glass slide. The dissecting scope in the center is used to magnify larger samples and is limited on the level of magnification. The last microscope on the right is an electron microscope, which uses an accelerated beam of electrons as a source of illumination and can magnify samples to much higher levels than the light or dissecting microscope. Here are some examples of images taken on a light microscope. The types of images range from cells to microorganisms and cross-sections of tissue. A modification of the light microscope called the fluorescent microscope can allow visualization of cells or tissue labeled with colors that identify specific components, like the two samples shown on the right-hand side of a cell going through mitosis and a cross-section of the hippocampus of a mouse being stained with a technique called rainbow. These images are examples of the type of visualization you would see on a dissecting microscope, where you would see full organisms or samples of individual organisms, but you would not be able to see cross-sections or smaller magnifications. These images are from two of the main types of electron microscopes. The one on the left is from the scanning electron microscope, which can be used to image the surfaces of samples, while the image on the right is from a transmission electron microscope, which can be used to image cross-sections of a sample in very fine detail. After the development of the microscope, many scientists were able to image what we now know as the smallest unit of life, the cell. From this, scientists were able to develop ideas, which we call the unified cell theory. This is proposed by Matthias Schleiden and Theodor Schwann in the early 1800s, and it states that, number one, all living things are composed of one or more cells. Two, the cell is the basic unit of life. And number three, all new cells arise from existing cells. In modern scientific standards, we classify all living cells into one of two groups, one being prokaryotic cells and two being eukaryotic cells. All of these cells share four common components, however. They all must have a plasma membrane, a cytoplasm, they have DNA, and ribosomes. The prokaryotic cell shown here is characterized by having no nucleus, relatively small in size, and no true membrane-bound organelles. The example shown here is a bacteria cell called E. coli, with most of its anatomy identified. Some of the most noteworthy are the chromosomal DNA, located in the region here called the nucleoid, and the flagellum over here, which is used for locomotion of the cell. There are many shapes and sizes of prokaryotic cells. Here are just a few. The first one here is a spherical bacterial shape called cocci. The first image on the left is taken using a light microscope, and the second image is using a scanning electron microscope. A staph infection is by a bacterium which has this type of spherical shape. The second shape is called bacilli, and the cell has more of a rod-shaped structure to it. And E. coli is an example of this type of shape. Here on the left you see a light microscope picture, and on the right a scanning electron microscope picture. There are many other shapes to bacterial cells that are noteworthy. One of them being an interesting shape that is a spiral shape shown here. This is a example of a bacterium that is in the group called Spirillum, and it is known that one of these types of species could be transmitted from rats to humans and could cause a disease called the rat bite fever. The second general type of cells is called the eukaryotic cell. We will study two groups of organisms that are made of eukaryotic cells, the first here being the animal. This is a drawing of a human cell, which has all of the major organelles identified. Functions of each we will discuss later in this lecture. The major characteristics to be noted here are the nucleus, the larger size compared to prokaryotic cells, and membrane-bound organelles. And in the animal cell, they have a specific organelle called the centrosome. Take some time to pause the video and review all of the animal eukaryotic anatomy listed here in this image. Okay, I wanted to show you one animal eukaryotic cell because all eukaryotic cells have various shapes depending on their functions. Here is an example of a neuron cell in a petri dish trying to connect to other neurons. Note the dynamic nature of the projections of the cell body as it's trying to make connections with other cells within that petri dish. It is migrating as it's moving along the bottom of the petri dish and constantly trying to make new connections with other cells. The second group of eukaryotic cells is the plant cell. This is a drawing of a fern cell, which has all of the major organelles identified. Functions of each we'll discuss later in the lecture, most of which will be identical to the functions in the animal cell. The major characteristics to be noted here are the nucleus, the larger size compared to prokaryotic cells, and the membrane-bound organelles, and in plants only, two other organelles called the chloroplast and the cell wall. Again, take some time to pause the video and review all of the plant eukaryotic anatomy. Plant cells are not as dynamic in shape as animal cells, but still have unique features. The video seen here shows living plant cells in an aquatic plant called Elodea. The movement seen is of chloroplasts, organelles actually moving around in the cell. These are the little green spherical shaped structures that you see here. This is called cytoplasmic streaming. Each cell is separated by the highly visible cell wall that you can see here. And as you can see, the shape of the cell does not change. As the components inside the cell move around, it is fairly rigid due to the structure of the cell wall, but yet there's some dynamic functions going on inside of the cell. Over the next few minutes, we're going to review all of the organelles and their functions in both animal and plant cells. So I would take some time to pause the video intermittently throughout this time to take notes on the names and the functions of these particular organelles. The first organelle we're going to talk about is the plasma membrane. It essentially separates the internal components from the surrounding environment of the cell. It is the gatekeeper. It lets things move in and out of the cell, and we will talk in more detail about the plasma membrane here in a few minutes. The second is the cytoplasm. And the cytoplasm is essentially the area between the plasma membrane and most other organelles in the cell itself, like the nuclear envelope. And essentially all organelles are suspended in this gel-like substance called cytosol. A lot of times scientists will use the words cytoplasm and cytosol interchangeable. The third organelle, called the cytoskeleton, is essentially a network of protein fibers, and it provides the structure to the cell and allows for a pathway of movement of vesicles within inside the cell to move along that particular cytoskeleton. The fourth main structure is called the endomembrane system, and it's not really a structure itself. It is actually made up of five different organelles within inside the cell itself. The first being the nucleus, which houses your DNA, the nuclear envelope, which is essentially a double membrane that houses all of the DNA of your nucleus. The rough endoplasmic reticulum, which is connected technically to the nuclear envelope and essentially modifies proteins. The smooth endoplasmic reticulum's job is to synthesize lipids that the cell may need for other functions. And then lastly, the Golgi apparatus, which will take modified proteins in the endoplasmic reticulum and sort, package, and distribute them along with some lipids to various locations within the cell or outside of the cell. The lysosome is a small vesicle that is basically the center with which digestive enzymes used to recycle components within inside the cell. These vesicles and vacuoles, these are basically sacs. They'll function in storage and transport. Ribosomes, these are structures responsible for protein synthesis. They can be connected to the rough endoplasmic reticulum or free-floating within inside of the cytoplasm. The mitochondria, that is responsible for making ATP, which is a molecule that is known for carrying energy and can help make other metabolic processes happen within inside the cell. Peroxisomes, these carry out reactions called oxidation reactions, and essentially they're used to break down molecules called fatty acids and amino acids. The cell wall, which is specific to the plant cell only, is an external structure which provides support for the cell. The chloroplast, also native to the plant cell, almost always. There are a few exceptions where animals will have a chloroplast, but they're very rare. These essentially, the chloroplast functions in photosynthesis and making sugars that the cell will use for making energy. The central vacuole is also another structure that's located in the plant cell, and it regulates the concentration of water in the plant cell. And the last two major organelles are these things called the extracellular matrix. This essentially is not really an organelle inside the cell, but instead it is made up of proteins and glycoproteins that essentially hold all of the cells together in a tissue structure and help with communication. And lastly, the intercellular junctions. These are essentially pathways in which cells will join together and sometimes will actually use communication between cells. There are several different kinds depending on how the communication is made and what kind of passageway is created between that junction. One of them, called the plasmadesmata, is a channel between two adjacent cells that's specific to plants. A tight junction, these join adjacent cells together, but it does not create a channel. The desmosomes join two animal cells together, so this is specific to animals and not plants. And then the gap junction is essentially indicative of a plasma desmata, but in animals, so it creates a channel between two animal cells. As promised, we're going to talk a little bit about the plasma membrane. And it needs to be studied in more detail. And the reasoning being it's the gatekeeper, so to speak, for things moving into and out of the cell. The modern model for the plasma membrane is called the fluid mosaic model, where the plasma membrane is described as a mosaic model made of components including phospholipids, cholesterol, proteins, and carbohydrates. Take some time to pause the video and review the model shown here so that you can find examples of phospholipids and all the rest of the components of the fluid mosaic model here. What I want to talk about now is there are multiple ways items can cross the plasma membrane. We group them into two main types, first being the passive transport. It does not require ATP or energy. ATP again standing for adenosine triphosphate, which we will come to learn a little bit more about in future chapters. The second type of movement is called active transport, and this does require energy or ATP. So with passive transport, which doesn't require energy, there are three main categories. The first being simple diffusion. This is essentially the movement of a molecule down a concentration gradient, meaning that it will move from a high concentration to areas where there is no or low concentrations of that particular molecule. Molecules that can do simple diffusion across the membrane need to be nonpolar and very small, so molecules like O2 are an example of that, or oxygen. The second type is called facilitated diffusion, and essentially this is diffusion across the membrane, but the molecule needs help to get across. And essentially we use a protein called a transmembrane protein, and it will create a channel which allows movement of the molecule down a concentration gradient. Again, meaning moving from a high concentration to a low concentration. The last type of passive transport is probably the most studied type of passive transport because it is actually looking at the diffusion of a specific molecule called water. And in fact, we look at how water moves across membranes when put into, a cell is put into three different types of solutions. The osmolarity of a cell, or the concentration of solutes in a solvent, the solvent being water, of a cell compared to its extracellular kind of fluid, can be categorized in these three types of solutions. Hypotonic, which means a very low concentration of solutes inside of the cell compared to outside of the cell. This will cause water to rush into the cell, and in animal cells will cause that cell to lyse. Hypertonic solutions are the exact opposite of that. You have very low concentrations of solutes outside of the cell compared to high concentrations outside of the cell compared to inside of the cell. And water will rush out of the cell to equalize those concentrations, and it basically shrivels the cell, and those cells will become misshapen and not function like they normally should. The third kind is called isotonic, An isotonic is essentially movement of water inside and outside, but in equivalent amounts. And this doesn't change the shape of the cell at all. It will not shrivel or lyse, kind of swell and lyse. It just creates a normal, functioning, homeostatic situation of the cell. The second kind of movement, called active transport, which requires energy, can be broken down into really three main groups for your level of class. The first one actually has the same name as the category, called active transport. And this essentially, you have another protein that creates a channel, but to open this protein and open the channel, an ATP molecule is required. And so every time a molecule passes through the channel, energy molecules like ATP are utilized or used up during that process. The last two kinds of active transport are called endocytosis and exocytosis. Essentially, these are where molecules can move in and out of the cell, but these are really large particles. These can be food or other organisms like bacteria that can be engulfed by the cell. And endocytosis meaning large particles move in, exocytosis meaning large particles can move out. Okay, so that is the conclusion of this chapter. If you've been paying attention, you should now be able to describe prokaryotic and eukaryotic animal and plant cell types, compare and contrast microscopes, summarize the cell theory, describe the fluid mosaic model, summarize the functions of cell organelles, and describe how the plasma membrane functions as a selectively permeable membrane. Now that you've looked at all the material in Chapter 3, there are a few things you could do next to help you succeed in learning this material. Take a look at the list shown and do as many as you can. Thank you for listening and look forward to seeing you in the next chapter.